Today, my partner, Heidi, and myself, Reina, are going to be talking about sickle cell disease. To understand sickle cell disease, let us first understand Mendel's first law of genetics, which is also called law of segregation. So first we have to understand some of the words, and the first word is phenotype, which is the physical appearance of a particular trait, and here we have blood cells, which one is sickly or curved and the other one is normal, which are the traits. For sickle cell disease. We also have recessive sickle cells. So sickle cells are recessive, and here we see that dominant is the normal cells because we have more of that. We see two alleles or two different alleles, which is straight hair and curly hair. Heterozygous means that an organism has two different alleles of a gene, and homozygous it means that it's a carrier of two copies of the same dominant allele, or not dominant, but two types. And here we have a Punnett square. The Punnett square tells us the combination of dominant alleles and recessive alleles. And here we are actually looking at the sickle cell. So pedigree is very important for genetic counselors and for us as well, because pedigree tells us if we inherited a disease from our parents. First analyze the key. Square is male, circle is female, all shaded in is sickle cell, half shaded in is carrier of sickle cell. So here we see Carlos, who is half shaded, which means that he's a carrier. Maria is all shaded in, which means that she has sickle cell. Eric doesn't have anything. Anna has sickle cell. Juanita is a carrier. Her husband is has sickle cell, and the baby has sickle cell as well. Ask yourself what causes sickle cell, and here are some of the causes: hanging out in the sun, gene mutation, viruses, and radiation from X-rays and all that kind of jazz. So here we're going to be focusing on gene mutation, and the gene mutation that causes sickle cell is called base substitution translation. And base substitution translation is a mutation where a single base is substituted by another base. So here we see G changing to T. So this is the DNA sequence, and G changes into T right here, and that causes the problem. So that's the base substitution. So we're going to be talking more about base substitution translation in depth, and we are going to be comparing the normal cell, the normal mutation with a sickle cell mutation. So we're going to compare and see how they change through each step. So first we have the DNA, which is a codes for something, and we have G. Oh, here it goes. So this is the normal mutation, the healthy mutation, the good one, and the DNA that the three letters the code for it is GAG, and then we have mRNA, which is CUC, and then we have our amino acid, which is glutamine acid, and these are polar and hydrophilic, and these are the good ones. Okay, that's the pair that actually makes a change for us to get sickle cell. And then for the sickle cell, we have GTG, that's DNA, and then the mRNA is CAC. So as you can see right here, that T was coded for something else. And then we have our amino acid, which is valine, and which is non-polar hydrophobic, and this is the bad one. So as you can see that these two pairs are different. So here are some of the symptoms of sickle cell disease. And the first one is tired. You feel very tired with sickle cell anemia. Dizziness, because you don't have a lot of oxygen. It's hard for you to breathe. Fast heart rate. And some of the treatments for sickle cell disease is are yeah, I spelled it wrong, but I corrected myself. Oxygen. You have to stay very hydrated when you have a sickle cell anemia. Yeah, painkillers, because uh, there are things that doctors may do to you that are very painful, like blood transfusion and yeah, and oxygen. So you may get oxygen, like oxygen tanks that have the mask on, so you can put it on, and that way it helps you to breathe easier. And the people that are at risk of getting this disease are African Americans, Central America, and South America. So those are the people.